This demo introduces the Neural Network Clustering tool, which can be used to group data with similar characteristics. This tool designs self-organizing maps to cluster input data and arranges the clusters so that similar clusters are adjacent to each other in the map. We will use a self-organizing map to cluster iris flowers based on their attributes. To start, we will open NNStart. From here, we can launch the clustering tool. The clustering tool can also be launched by typing nctool at the command line or from the MATLAB Start menu. The first panel of the clustering tool shows a diagram of a self-organizing map. It consists of a single layer of neurons arranged in a two-dimensional grid. We will train the map so that every neuron responds to a different cluster of flower features, and adjacent neurons represent similar clusters. The second panel allows us to select problem data from the MATLAB workspace, or load an example dataset. We will select the iris flowers dataset, which contains a 4 by 150 matrix defining 4 attributes of 150 different iris flowers. We will try a 6 by 6 grid of neurons, which results in 36 clusters. Now we will train the self-organizing map. The topology plot shows the 36 neurons arranged in a 6 by 6 two-dimensional hexagonal grid. The sample hits plot shows how many of the training samples fall into each of the clusters. Adjacent clusters have learned to classify similar flowers. The gap with no hits is an indication of a separation between clusters with no intermediate flowers. The neighbor distances plot shows the 36 neurons with blue hexagons. The distances between the neighboring neurons are represented with bright colors between the neurons whose clusters are similar, and dark colors between clusters which are farther apart. Bright regions indicate a smooth distribution of flower features. Dark borders indicate that few or no flowers fall between those clusters. The input weight planes plot shows the weights from each of the 4 flower features to each of the 36 neurons. Weight planes that look similar indicate features which are highly dependent, while dissimilar planes indicate independent features. The weight position plot shows the flower samples as green dots in terms of their first 2 features. Then the neurons are plotted as dark blue dots, according to their first 2 weights. The red lines indicate which neurons are neighbors. Note how the neurons have spread out, with neighboring neurons representing adjacent areas of the flower feature space. The two-dimensional map looks folded in places in the plot, because it is really spread over 4 dimensions of flower features. The final panel lets us generate a sample MATLAB script. Here we define the input data. Next, we create the self-organizing map, which we then train. Once trained, we can calculate the network's outputs for any inputs, view the network diagram, and create any number of optional plots. We can also save our results to the MATLAB workspace. This concludes the demonstration of the Neural Network Clustering tool. You can view other demos to see how to solve other types of problems with neural networks.